This talk is about dismantling the fragility index. So the fragility index has been introduced as a complement to the p-value to summarize the strength of statistical evidence for a trial. It's defined in trials with binary or time to event outcome and two equal size arms, and it's calculated as the minimum number of swaps from non-event to event in the treatment arm needed to shift the p-value from Fisher's exact test over 0.05. So here's an example of a malaria vaccine trial with two equal size arms and binary outcome. And in order to calculate the fragility index, you take people who experienced an event in the treated arm and swap their status from event to non-event and count how many swaps are needed to get the p-value from Fisher's exact test over 0.05. So the fragility index for this trial is 2, because it took 2 swaps to get p over 0.05, and lower values of the fragility index indicate higher fragility. This tool is intended to complement the p-value and help clinicians interpret how robust their results are, and it's been applied in dozens of review papers in various fields to characterize robustness of results amid concern over reproducibility of research. Here's an example of 2 hypothetical trials. They have different sample sizes, equal p-values, and different fragility index values, and have low risk for bias. This example was provided by Walsh et al. To give the reader an intuition for the concept of fragility. So it represents the conceptual foundation of the fragility index. What you can see is that the smaller trial, in order to match the p-value of the larger trial, it needs to have a larger effect size. In this case, the effect size is measured as 1 minus the relative risk. You can also see that the confidence interval of the smaller trial is farther away from the null value of 0. But its fragility index is smaller, indicating it's more fragile. So the authors write, the fragility index of only 1 would suggest that caution is warranted in interpreting the results of the small trial. So the authors had explicitly stated these 2 trials have low risk for bias, and I thought it was interesting to consider, what if we had 2 trials that didn't have any bias? This is interesting for 2 reasons. Number 1, it can help us understand by exploring mathematically which trial has more statistical evidence. It helps us understand from a perspective of probability what's going on and whether this fragility index is capturing something different from the p-value. Number 2, you might think that in real life trials always have some bias, but it's possible to imagine 2 early phase trials that don't have any missing data and don't have any dropout, but they have different sample sizes and the same p-value. The smaller trial has to have a larger effect size than the larger trial, but it's more fragile. So the question is, which of these 2 different treatments is more likely to work? So if we want to explore this from a perspective of probability, we can start out by looking at the probability statements that define the p-values. In the smaller trial, the p-value of 0.02 is the probability of observing a treatment effect, a test statistic as or more extreme than what was observed, assuming there's no treatment effect in a trial with 100 per arm. And in the larger trial, it's the probability of observing a test statistic as or more extreme than what was observed, assuming no treatment effect in a trial with 4,000 per arm. So the probability statement defining the p-value conditions not only on the null hypothesis, but also on the sample size. Those 2 probability statements don't have the same meaning. P equals 0.02 has a different meaning in a small trial than in a large trial. So which trial has more evidence statistically? Well, it's been studied before in the context of testing a population mean by analyzing the posterior probability of a treatment effect, which is, more scientifically interesting and relevant than the p-value. Richard Royal studied it for comparing 2 point hypotheses. Berger and Selke studied it for a point null and a composite alternative, and I extended their methodology to the 2x2 design. So I'm going to explain the methodology to you for a single proportion. So for testing a single proportion, we have a coin toss experiment. We toss a coin n times and use the number of heads to test whether the coin is fair. So suppose you have 2 different trials, 2 different coins, A and B. Coin A had 10 heads and 10 tosses. Coin B had 66 heads and 100 tosses. They have the same p-value, but coin A has a much larger effect size and confidence interval farther away from 50%, but it's more fragile. So which coin do you think is more likely to be unfair? Okay, so we're going to study this using Bayes' rule, and here are the hypotheses that we're testing. Bayes' rule allows us to factor the posterior odds into the prior odds times the Bayes factor. The denominator of the posterior odds is just 1 minus the numerator. That's why this is the posterior odds. And we're going to start out assuming a 1-to-1 prior odds, in which case the posterior odds is equal to the Bayes factor. The denominator here, probability of number of heads for a fair coin, that's just a binomial distribution. For the numerator, probability of probability of number of heads given an unfair coin, for that, we need to impose a prior on this parameter space and integrate over it. So I imposed a uniform prior. When you integrate over that, you get a discrete uniform, which makes sense if your prior belief about the probability of heads is equally likely anywhere between 0 and 1. It makes sense that the distribution for your number of heads is equally likely anywhere between 0 and sample size. Here are the null and alternatives distributions on top and bottom respectively for the small and large trial. The Bayes factor is the ratio of yellow to red. It's larger in the small trial. That's because in the large trial, the alternatives distribution is flattened over a much larger set of points. From the Bayes factors, we can calculate the posterior probability with a 1-to-1 prior odds. So if you thought that 10 out of 10 heads is more likely to be an unfair coin, you're right. But the finding that the smaller trial has more evidence is independent of the choice of prior odds. So the smaller trial has a larger Bayes factor than the large trial. So whatever ratio we multiply it times here, the smaller trial is going to have a larger posterior odds and therefore posterior probability than the larger trial. I repeated the analysis for the 2x2 design where we're comparing two proportions instead of testing a single proportion. I generated a bunch of contingency tables that have the same p-value and progressively larger sample sizes. So for example, this contingency table represents 55 events out of 100 people in one arm and 36 out of 100 people in another arm. It has Fisher's exact test p-value equals 0.01. And then using the same set of assumptions, just extending them to 2x2 design, I calculated these posterior probabilities. So again, you see for a fixed p-value, the smaller trial has more evidence for a treatment effect than the larger trial. And the Bayes factors follow the same pattern. So this pattern is, or the finding is independent of the choice of prior odds. So if two trials have the same p-value and different sample sizes, the smaller trial has more evidence for a treatment effect as measured by the posterior probability. But the fragility index says the opposite, and this could lead to incorrect decision-making. So if you think of the example with two early phase trials with no bias, and they have the same p-value, different sample sizes, the probability that the treatment in the smaller trial, considering all the evidence, works is larger than the posterior probability of the treatment in the larger trial. So we should move forward with the treatment tested in the smaller trial. And the fragility index would lead the investigator to make the opposite decision. So it's promoting an incorrect intuition of probability. And part of why that happened is because a mathematical exploration of its properties was not performed before it was promoted as a tool. The fragility index has a number of other problems, which I don't have time to go to in this talk. But because of those problems, there have been offshoots of this index that have been proposed to kind of patch over those problems. For example, a modified index accounting for times in survival analysis data, choosing a predefined threshold for fragility, in other words, dichotomizing it, dividing it by the sample size, etc. But just like with the fragility index, the operating characteristics and mathematical properties of these offshoots haven't been investigated. So there's not really been an exploration of how they actually are going to perform in terms of informing decision making. So until that's done, I would be reluctant to recommend these either. I wanted to thank several people who provided comments on these work and give you some references. Thank you.